Class 11, Unit 4, Plant Physiology. Melvin Kelvin. Melvin Kelvin, born in Minnesota in April 1911, received his PhD in chemistry from the University of Minnesota. He served as professor of chemistry at the University of California, Berkeley. Just after World War II, when the world was under shock after the Hiroshima Nagasaki bombings and seeing the ill effects of radioactivity, Kelvin and co workers put radioactivity to beneficial use. He, along with J.A. Bussum, studied reactions in green plants forming sugar and other substances from raw materials like carbon dioxide, water, and minerals by labeling the carbon dioxide with carbon 14. Kelvin proposed that plants change light energy to chemical energy by transferring an electron in an organized array of pigment molecules and other substances. The mapping of the pathway of carbon assimilation in photosynthesis earned him a Nobel Prize in 1961. The principles of photosynthesis, as established by Kelvin, are at present being used in studies on renewable resource for energy and materials and basic studies in solar energy research. Chapter 11, Photosynthesis in Higher Plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have already learned that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are called heterotrophs. Green plants carry out photosynthesis, a physiochemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. It is the primary source of all food on Earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transform light energy into chemical energy. 11.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, that is green pigment of the leaf, light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light. Another experiment you may have carried out where a part of a leaf is enclosed in a test tube containing some KOH soaked cotton which absorbs CO2 while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of starch later in the two parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch, while the portion that was in the tube tested negative. This showed that CO2 was required for photosynthesis. Can you explain how this conclusion could be drawn? 11.2 Early Experiments. It is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. Joseph Priestley, 1733 to 1804, in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, such as a bell jar, soon gets extinguished, figure 11.1 A, B, C, D. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove, that is, oxygen. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, Jan Ingenhousz 1730 to 1799 showed that sunlight is essential to the plant process that somehow purifies the air fouled by burning candles or breathing animals. Ingenhousz, in an elegant experiment with an aquatic plant, showed that in bright sunlight some bubbles were formed around the green parts, while in dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is only the green part of the plants that could release oxygen. It was not until about 1854 that Julius von Sack provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. His later studies showed that the green substance in plants, that is chlorophyll as we know it now, is located in special bodies. These special bodies were later called as chloroplast within plant cells. He found that the green parts in plants is where glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiments done by T.W. Engelmann 1843 to 1909. Using a prism, he split light into its spectral components and then illuminated a green alga, that is cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the site of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B discussed in section 11.4. By the middle of the 19th century, the key features of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as CO2 plus H2O in the presence of light gives CH2O plus O2, where CH2O represented a carbohydrate, example glucose, a 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist Cornelius van Niel 1897 to 1985, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by the equation 2H2A plus CO2 in the presence of light gives 2A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis. When H2S instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not O2. Hence, he inferred that the O2 evolved by the green plant comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore as follows: 6CO2 plus 12H2O in the presence of light, it gives C6H12O6, that is glucose, plus 6H2O plus 6O2, where C6H12O6 represents glucose. The O2 released is from water. This was proved using radioisotope techniques. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? 11.3 Where does photosynthesis take place? You would of course answer in the green leaf or in the chloroplast. Based on what you earlier read in chapter 8, you are definitely right. Photosynthesis does take place in the green leaves of plants, but it does so also in other green parts of the plants. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplasts will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is a membranous system consisting of grana, the stroma lamellae, and the matrix stroma, figure 11.2. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn forms starch. The former set of reactions, since they are directly light driven, are called light reactions, that is, photochemical reactions. The latter are not directly light driven but are dependent on the products of light reactions, that is ATP and NADPH. Hence, to distinguish the latter, they are called by convention as a dark reaction or carbon reactions or light dependent reactions. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. 11.4 How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves even in the same plant? We can look for an answer to this question by trying to separate the leaf pigments of any green plant through paper chromatography. A chromatographic separation of the leaf pigments shows that the color that we see in leaves is not due to a single pigment but due to four pigments: chlorophyll A, which is bright or blue green in the chromatogram; chlorophyll B, which is yellow green; xanthophylls, that is yellow; and carotenoids, that is yellow to yellow orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelength. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A pigment to absorb lights of different wavelengths, figure 11.3a. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the VIBGYOR. From figure 11.3a, can you determine the wavelength, that is color of light, at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelength too? If yes, which one? Now look at figure 11.3b showing the wavelength at which maximum photosynthesis occurs in a plant. Can you see that the wavelength at which there is a maximum absorption of chlorophyll A, that is in the blue and red regions, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 11.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? Its answer is yes. These graphs together show that most of the photosynthesis takes place in the blue and red regions of the spectrum. Some photosynthesis does take place at the other wavelength of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophylls, and carotenoids, which are accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis but also protect chlorophyll A from photooxidation. 11.5 What is light reaction? Light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates ATP and NADPH. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes LHC within the photosystem 1 PS1 and photosystem 2 PS2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC, that is light harvesting complex, are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments except one molecule of a chlorophyll A, forming a light harvesting system also called antenna, figure 11.4. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both photosystems. In PS1, the reaction center chlorophyll A has an absorption peak at a 700 nanometer, hence is called P700, while in PS2 it has absorption maxima at 680 nanometer and is called P680. 11.6 The electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor which passes them to an electrons transport system consisting of cytochromes. Figure 11.5. This movement of electrons is a downhill in terms of an oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of photosystem PS1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy-rich NADP+. The addition of these electrons reduces NADP+ to NADPH plus H+. This whole scheme of transfer of electrons starting from the PS2 uphill to the acceptor, down the electron transport chain to PS1, excitation of electrons, transfer to another acceptor, and finally downhill to NADP+, reducing it to NADPH plus H plus is called the Z scheme, क्योंकि Z की तरह symbol बन रहा होता है due to its characteristic shape. Figure 11.5. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. 11.6.1 Splitting of water. You would then ask, how does PS2 supply electrons continuously? The electrons that were moved from photosystem 2 must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS2. Water is split into 2H plus and nascent oxygen and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2 via the help of splitted water. 2H2O gives 4H plus O2 plus 4 electron. We need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and O2 formed likely to be released, in the lumen or on the outer side of the membrane? 11.6.2 Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. The process through which ATP is synthesized by cells in mitochondrial and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS2 and then the PS1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADP H plus H plus are synthesized by this kind of electron flow, figure 11.5. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons, figure 11.6. A possible location where this could be happening is in the stroma lamellae. While the membrane or lamellae of the grana have both PS1 and PS2, the stroma lamellae membranes lack PS2 as well as NADP reductase enzyme. The excited electrons does not pass on to NADP plus but is cycled back to the PS1 complex through the electron transport chain, figure 11.6. The cyclic flow hence results only in the synthesis of ATP but not of NADPH plus S plus. Cyclic phosphorylation also occurs when only light of wavelength beyond 680 नैनोमीटर are available for excitation, क्योंकि यहाँ पर हम लोग P700 वाला ही फोटो सिस्टम यूज़ कर रहे हैं.